Hey everyone, welcome back to The Explainer. Today, we're diving into a fascinating scientific journey to solve what was honestly one of the most frustrating biological mysteries in medical history. We are talking about leprosy. It's a disease that's literally haunted humanity for millennia, and we're going to explore the incredibly unique and frankly kind of bizarre ways the animal kingdom eventually stepped up to help us understand and fight it. So imagine trying to cure a disease that absolutely refuses to be studied. Mycobacterium leprae is actually the very first pathogen ever identified as causing disease in humans. But researchers hit a massive brick wall right out of the gate. It flat out refused to grow in a petri dish. No matter what artificial medium scientists cooked up in the lab, these bacteria just wouldn't multiply. Zero. Zilch. And without being able to cultivate it, figuring out how it works or testing new drugs was virtually impossible. This forced scientists to completely ditch the petri dish and hunt for a living laboratory instead. Here's how we're going to break down this incredible story. We'll start with the petri dish problem, then the mouse breakthrough. Next, enter the Nude mouse, followed by the Armadillo factory. After that, primate discoveries. And finally, we'll wrap up with the verdict. Let's get right into it. Section 1. The petri dish problem. Looking at the timeline of events, you can see a staggering 86-year gap of totally failed experiments. Just think about that for a second. Over 8 decades between our Hansen discovering the bacteria back in 1873 and someone finally figuring out how to actually grow it. Researchers tried infecting monkeys, cats, rabbits, the whole shebang, with almost zero success. But then, in 1956, a scientist named Binford had a real light bulb moment. He noticed that human leprosy lesions usually popped up on cooler parts of the body, like the ears, and hypothesized that the bacteria actually preferred cooler temperatures. That single observation was an absolute game changer. It paved the way for Shepard in 1960, who finally succeeded by targeting the coolest part of an animal he could find, the mouse footpad. Since artificial media was a complete bust, scientists had to create this strict Goldilocks rubric for the perfect animal model. It was essentially a massive biological scavenger hunt. They needed an animal that was immunologically intact, but still susceptible enough to show the full clinical spectrum of the disease. It had to develop peripheral neuritis, which is that devastating nerve damage leprosy is known for. Ideally, it also needed to be genetically close to humans, live long enough to study such a slow-moving disease, and, not break the bank while being easy to keep in a lab environment. Finding a creature that actually ticked all of these boxes, yeah, that was no small feat. Section 2. The mouse breakthrough. Riding on that cooler temperature hypothesis we just talked about, Shepard infected the footpad of a normal, healthy mouse. And wow, did it work. The mouse footpad suddenly gave us a super efficient in vivo method to screen new anti-leprosy drugs and even test for drug resistance. For the very first time, researchers could actually give a drug to a living subject and definitively measure if it stopped the bacteria from multiplying. It was a massive step forward. But as always in science, there was a catch. The thing is, normal, healthy mice have T cells that eventually step up and fight back. In immunocompetent mice, the bacteria multiply to about 1 million, but then the normal immune system just slams on the brakes. The infection stays right there in the footpad with absolutely no systemic spread. Now, that's great for the mouse, but to study severe disseminated leprosy, the kind that completely ravages the whole body, scientists realized they desperately needed a model without those specific immune defenses. Section 3. Enter the Nude mouse. Introduced to leprosy research in 1975, the Nude mouse is a highly specialized biological tool carrying a natural congenital mutation. And yes, they are completely hairless, which is where they get the name Nude. But the much more critical detail here is that they are athymic. They only have a rudimentary thymus, which means they literally cannot produce the T cells that would normally stop the leprosy bacteria in its tracks. So without an immune system to keep things in check, the bacteria absolutely thrived. This resulted in massive infections, dramatically enlarging the Nude mouse's footpad. The bacteria multiplied wildly and spread all the way to the liver and spleen. Because the infection was allowed to run completely uninhibited, the Nude mouse became this unparalleled model for studying leprosy neuropathy, which is the nerve damage, and for running really crucial long-term chemotherapy trials. Section 4. The Armadillo factory. Now, let me introduce you to the 9-banded armadillo. This primitive, burrowing mammal quickly became the second most extensively studied model in all of leprosy research. Why an armadillo of all things?
because it was the very first immunologically intact species we found that could actually develop full-blown disseminated lepromatous leprosy.
It turns out this odd little creature had the exact, highly specific biological traits we were desperately looking for. First off, its core body temperature naturally hovers right around 30 to 35 degrees Celsius. And remember what we learned, that is the absolute sweet spot for Mycobacterium leprae. Secondly, they have a solid 12 to 15-year lifespan, which gives scientists plenty of time to study the slow progression of the disease. Oh, and incredibly, they give birth to monozygotous quadruplets. That means 4 genetically identical babies every single time, which is an absolute dream scenario for controlled scientific experiments. They were basically perfectly designed natural incubators for the disease. And when we say incubators, we really mean it. To put this into perspective, a single infected armadillo can produce up to a staggering 1 trillion bacilli. 1 trillion! That astronomical number finally allowed the World Health Organization to harvest enough bacteria to kickstart massive immunology programs. But, plot twist, scientists later discovered that armadillos actually carry the disease naturally in the wild. They are a natural reservoir, meaning they can pass the disease right back to humans, which obviously throws a huge wrench into our global efforts to eradicate leprosy once and for all. Section 5. Primate Discoveries. Eventually, to really understand how the disease impacts humans, researchers had to look to our closest phylogenetic cousins. They tested several primates. Chimpanzees did develop self-healing lesions, but they didn't quite mimic the severe human form of the disease. The Setimunga monkey, however, that was a massive breakthrough. Researchers discovered that these monkeys acquire lepromatous leprosy naturally, and they exhibit symptoms that are strikingly, almost disturbingly, similar to what human patients experience. Unlike the mice or the armadillos, primates display the severe, highly visible clinical spectrum of the disease. They develop heavy facial involvement, distinct nodules, and even ocular or eye involvement. These monkeys basically provided a vital window into the systemic, visible ravages of leprosy that rodents just couldn't show us. While it's certainly tough to look at, this definitively proves their immense value to medical research. These monkeys actually develop severe clawing of the hand. This is directly caused by peripheral neuritis. That's the exact same nerve damage that tragically afflicts human patients and leads to such severe disability and loss of motor function. Being able to study these primates finally allowed researchers to decode the mechanics behind leprosy's most devastating physical impacts. Section 6. The verdict. So, looking at our final summary matrix, how do they all stack up? Well, it's pretty clear that no single animal turned out to be the magical Goldilocks model. Normal mice are awesome for cheap, easy drug screening, but they don't yield much bacteria. Nude mice are fantastic for chemotherapy studies, but their lifespan is just too short. Armadillos? They give us massive amounts of bacteria for vaccines, but they're incredibly hard to breed in captivity. And chimpanzees show those crucial human-like symptoms, but keeping them in labs is very difficult. So, instead of finding one perfect animal, researchers have to rely on this complex patchwork of specific advantages across multiple species to really understand and treat the complex manifestations of leprosy. The sheer ingenuity required by scientists to overcome that initial Petri dish problem is just astounding, isn't it? The incredible progress we've made against leprosy over the years proves that sometimes the key to human survival isn't found sitting in a glass test tube. Sometimes it's hidden in the strangest corners of nature, from a hairless mouse to a wild burrowing armadillo. It really leaves us to wonder, as we face down the complex pathogens of our future, what other seemingly impossible medical breakthroughs are just waiting to be uncovered in the animal kingdom?